Could you give me a little bit about your personal background and your professional background and how you got interested in the universe of sarcomas?
Sure. Yeah, it's sort of a convoluted story. I actually started out as an orthopedic surgeon. So I'm from New York originally. Went to medical school in New York as well, and then went to Yale for orthopedic surgery. I can say that I think I realized probably sooner than I would admit that I wasn't as captivated by clinical medicine as I was by research. And really pathology. Pathology was probably the thing I loved the most in medical school. It was not an especially popular field at the time, and it wasn't something that people were really encouraged to go into. So I sort of went through with my plan, which was orthopedic surgery. And I love orthopedic surgery. I still do now. But I think when I realized that I wanted to make a move outside of surgery, pathology was a very obvious choice to me. So I stayed at Yale to do my pathology residency. And I think, with my background, bone and soft tissue tumors were just a natural interest for me. When I started my pathology residency, it was sort of my goal to learn as much as I could about those. I was lucky that I had good mentors in both adult and pediatrics at Yale, pathologists who had specialized in sarcoma pathology. So I sort of clung to them as soon as I started my residency. I enjoy all of surgical pathology, but for sure, bone and soft tissue tumors were my primary interest. So after training, I did one more year of fellowship at Yale in GI and liver pathology, but then I went to Mayo Clinic and did another year fellowship there in surgical pathology and bone and soft tissue pathology. And I think really that was sort of just an immersive fellowship in bone and soft tissue, and that was a fantastic year. I spent time with Krishnan Uni, who's, at least in my opinion, I think in most people's opinion, sort of the world's preeminent bone pathologist, as well as Andrew Fulp, who is a world famous soft tissue pathologist. And again, I just really clung to those physicians and really learned everything that I could. And Mayo Clinic is a wonderful place for exposure to bone and soft tissue tumors. As you know, and as the Paula Takacs Foundation has emphasized as well, these are generally rare tumors. So in most places, it really is hard to get sufficient exposure. I think in most training programs, it's really hard to come by. So you really have to seek out big centers in places, generally they're referral centers, places that have significant volume bone and soft tissue, for you to really be able to get the exposure and develop any expertise. So I saw good material at Yale. I saw very good material at Yale. I saw fantastic material at Mayo Clinic, both the in-service, the Mayo Clinic specific, but also they have this incredible consultation service where we received cases from all over the world. And so we just did that all day, every day. So it was a fantastic year. And I'm very grateful for that experience.
Is that the typical trajectory from being an orthopedic surgeon to go into pathology, or is that more unusual?
I think it's very unusual. Orthopedics is a fantastic specialty, and people don't really leave orthopedics. Generally very happy when they go there. And in many ways I was as well. I did not have sort of that fire to go to the operating room every day. I didn't see that as being sort of my future. I was always very interested in research. And I think for me, I wanted to spend my time in a specialty where research was emphasized, and for me pathology was perfect for that. So I think I've met one other person actually who was in orthopedic surgery residency and then they left and went to pathology as well. So there's at least one other person like me, but generally people are very happy to be orthopedic surgeons and don't make the switch very often.
That's interesting.
Yeah.
One of the things that was so critical when Paula, Takacs, and I went to Levine Cancer Institute back in 2012-13, when it was a new cancer center, and we met with Dr. Edward Kim at the time and said to him, we are very, very interested in funding different types of research here at the Institute, basic science research and translational research and clinical trials, and you know, we were so blessed that it has come to fruition when you fast forward the tape to today. And you know, although it's in its infancy, you know, in the large picture, it's still rock solid and on its way. And there was many considerations, Dr. Kim said, as to why he said yes to us back then. And one of the major reasons that Dr. Kim said yes, we are interested in this endeavor, is you and your passion for sarcoma tissue collection and documenting the science of all these biopsy samples. Could you talk a little bit about, or a lot of bit about, how that happened for you and what your thought was and why you're so proud of the collection today?
Sure, I'm happy to. I came here in 2007, so I finished up in Minnesota at Mayo in June of 2007, started here in July. At the time, this was before Levine Cancer Institute, obviously. It was the Blumenthal Cancer Center at the time. We had a great group of people here already. So specifically Jeff Niesel, an orthopedic oncologist, Josh Pat, his younger partner, who's not so young anymore. Now that's a younger partner. We had Mike Livingston in surgical oncology. We had a great group of people from radiation oncology. So there was an active group when I got here, my group recruited me specifically to work with Jeff and Mike Livingston, because the person who was doing sarcoma pathology was retiring. So they brought me here with the express purpose that I was going to work with the team. And I thought, for me, the thing that I could bring to the table, I mean, obviously, I hope that I can bring diagnostic expertise. Of course, primarily that was what I was coming for, is to diagnose the patients correctly. But I had always hoped that we had material here. I mean, Charlotte is unique in that it's not New York City or it's not Los Angeles or Boston, these places where it's not all that surprising that you have very active sarcoma programs. I mean, it's a smaller city, not a small city, but it's smaller, but we still do have a really significant volume of bone and soft tissue tumors. So that material was there. So when I say we have a significant number, the number of cases that we see still really pales in comparison if you're talking about breast tumors or lung tumors, colon tumors, things like that. So that it's still not an overwhelming number of cases. And that is really a challenge when you're trying to do research and trying to investigate sarcoma. It's getting a number of cases, enough cases to investigate where any of the conclusions that you would make have any relevance. I mean, if I say we make an observation, 1 or 2 tumors, that's wonderful, it's interesting, but you know, I think most medical oncologists would be a little bit reluctant to change practice or give a drug or give radiation on the basis of sort of my anecdotal experience. So for me, when I got here, and partly it was for my own education, my goal when I started was really just to look at all the cases that had ever been seen here, going back into the 70s, look at those slides and just again, for pure experience sake. And then I did want to sort of follow them to know we've seen X number of osteosarcomas, or we've seen X number of liposarcomas. And you learn quickly enough that sometimes you see many specimens from a single patient, and Paula Takacs is an example of that. I think Paula was probably one of the first cases that I diagnosed here back in the day, and then over the years, she had sort of ups and downs with her disease, and we saw, unfortunately, we saw resection specimens from her multiple times, and that's frequently the case. But I think that keeping that material on site and keeping that material available, because at the time, so obviously 2007 isn't when genomic analysis started for tumors, but things were sort of taking off in down multiple avenues in investigating these tumors. And so, again, my goal was to keep them here and have them available, sort of annotate them as closely as I could, so that we could make connections between what these tumors look like on a slide to me, or what they look like on imaging, or clinically to Jeff or Josh, and then really do translational research with them. So it's helpful for pathologists that you can sort of make that connection, but I think ultimately, for me, my goal was always to try to contribute to the treatment of these tumors. It's hard to get excited about being the world's greatest diagnostic pathologist if every single one of your patients dies of their disease. So my goal was always to try to help Mike Livingston, to help Jeff Niesel, and my prediction was, and thankfully, Derek Ragavin came to town, Ed Kim came to town, researchers came to town, so it took a few years, but once Levine Cancer Institute launched, and it was really serendipity with the Paula Takacs Foundation that we had the support from the foundation that we have, everything really just sort of gelled. We had the material, we had the clinical expertise, the investigators came, and so that was our moment to strike and start doing translational research on these. So, again, I was lucky to end up in a place with the material and the physicians to work on that, and then extraordinarily lucky when the Levine Cancer Institute launched and brought in more people to work on those. So it's been a fun journey. Like you say, we're sort of primed and working hard now, but we're still really at the beginning of things. So there's still a tremendous opportunity to work on these tumors.
Sure. And as it's been explained to me, that to have all those tumor samples, but not to have the clinical annotation would be a big disadvantage. And in this case, it's so phenomenal that you have so much clinical information on top of. Is that correct to say?
Yeah, I think so. I think it's certainly a help. I think part of the challenge with working on these is it's sort of sarcomas are a very heterogeneous group of tumors. There's many different types. Mm-hmm. And I'll go out on a limb and say that I'm diagnosing these correctly, so that's good, because sometimes that is challenging to get the correct diagnosis. And then sort of keeping them in one place. I think that it's helpful to have. I built a database, and basically every single case that I saw of any interest to me educationally or down the road I thought with research, I entered in a database. So I have over 6,000 entries in that database now by diagnosis. And so if there's a specific research question, if someone, for instance, is interested in liposarcoma, a specific question, or synovial sarcoma, I'm able to say with that database, we have, 120 osteosarcomas that have been diagnosed here over the years, and those 120 patients then represent X number of biopsies, resections, re-resections, etc. So when there's a an idea on the table, and we have it, with my database, we have the ability to say, we have good material in this specific type of tumor. We know we have X amount of cases that we can test, and with the investigators that we have now, and with the machinery that we have here, I mean, even cases that are quite old, we still manage to get DNA from, we still manage to get RNA from those tumors. So really, it's a tremendous opportunity, and as we're going forward, I mean, the way we investigate these is ever more sophisticated. Nuri Steuerwald is a perfect example of someone who I think has been really instrumental in this effort with her knowledge. Her, Carol Farhang-Far, and again, sort of bringing the various technologies to the campus here.
Could you elaborate a little bit on that for our supporters?
Sure. So the goal or the challenge really is to try to identify specific aspects of these tumors that allow them to be treated. So again, I'm a diagnostic pathologist, so for me, I'm starting way upstream in making the diagnoses. So there's certainly value in trying to identify either new techniques or new markers to help diagnose patients. Again, as I mentioned before, really what we're all working towards is giving Mike Livingston and Ed Kim, Niesel, the tools to treat these patients. So that's really what we're trying to do with some of the genomic techniques, is to identify specific changes, specific alterations in the tumors. So basically, you're sort of exposing a target, because what you want to do, and as I think most people have probably some connection to, you have either yourself or your family member has had cancer and has to have been treated with chemotherapy or radiation. They are not gentle drugs to give people, and I think a lot of that is because really you're not able to directly treat the tumor. Unfortunately giving sort of toxic medications to a patient, and they're killing the tumor for sure, but they're having a tremendous effect on the patient as well. Generally, so people lose their hair, people have the nausea and vomiting, all the side effects that go along with chemotherapy. So what we're trying to do with this genomic investigation, next generation sequencing, et cetera, RNA sequencing, et cetera, is trying to identify, again, specific targets in those tumors, because if you can develop a drug or identify a drug that's already been being used that can just target that tumor, you're really looking for that silver bullet. So you can just kill that tumor, kill the tumor cells, whether they're in the tumor or whether they're already circulating in the patient's bloodstream. That's really the goal. You want to kill the tumor, but not harm the patient, not kill the patient. So that's our focus. Really, I mean, that's the primary goal, is to really figure out what makes these tumors unique, what gives them the advantage, how do they grow so quickly, how do they spread so quickly, are there other ways to diagnose them, ways that aren't as invasive. I mean, certainly nobody wants to have a biopsy or even an operation to take a biopsy, so we're looking at different means, testing blood, looking for tumor DNA in blood, things like that. So less invasive ways to diagnose, hopefully less harmful ways to treat. So that's sort of all of our activity right now is focused on that goal.
Have you seen a lot of momentum and advancement in science in the blood biopsy realm?
So I have seen some momentum and some advances in other solid tumors. I think, like, sort of coming back to the point that I made before, is that sarcomas are very rare. They frequently don't get the attention that higher profile tumors get. So, again, tumors that are very common, where there are a lot of people who are affected by it, they're frequently, and unfortunately, if I'm being honest, pharma is probably going to focus on a very common tumor more quickly than they would on a tumor that's extremely rare. There's incentives to do that, which are obvious. So I think that the research world, whether it's doing next generation sequencing or really any kind of research, sarcoma is always a few years behind the other solid tumors, because I think that there aren't as many people working on it. And I think the technology usually reaches. So there is really just over the past year, 2 years, been an effort to try to look at the circulating DNA, correlation with outcomes, with recurrence, et cetera, metastasis in sarcoma patients. But that's something that we're talking about right now in LCI, and I expect that that's something that we're going to be looking at here really quite soon, because it's not low-hanging fruit, but it's just an opportunity that you can't pass up, because I think you can probably convince a patient to give some blood a lot faster than getting a lung biopsy or getting a bone biopsy, or I think it's much easier to get people excited about testing blood rather than other biopsy tissue.
Absolutely.